Hello everyone, and welcome to our presentation on thyroid peroxidase by Chloe, Malika, Devin, and Claire. We've probably all heard of the thyroid gland. It's a butterfly-shaped tissue located at the base of the front of the neck. The purpose of this vital gland is to regulate body functions including metabolism, growth, and the development of the body. The thyroid gland does this by producing and releasing hormones. A steady amount of hormones being released is important because it can alter proper function of the body, which means it alters metabolism and causes health problems. Remember, metabolism is the process that transforms food into energy. If too many hormones are released, it can speed up metabolism more than normal. On the other hand, if too few hormones are released, it can slow down metabolism too much. Either of these cases can cause health problems. The hormones produced are called thyroxine, or T4, triiodothyronine, or T3, and RT3. Normally, approximately 80% of the thyroid hormones produced are T4 and 20% are T3. In order for the thyroid gland to produce thyroid hormones to maintain metabolism, it has to produce an enzyme called thyroid peroxidase, which is also known as TPO. Thyroid peroxidase helps with this process by assisting in a chemical reaction that produces the thyroid hormones, and this all occurs within the thyroid gland. The process of hormone synthesis, with the help of thyroid peroxidase, requires iodide ions. Iodide is present in many foods, such as dairy, eggs, and salt. After ingesting foods containing iodide, it gets transported into the plasma of the bloodstream. The first step of this process is for iodide ions to enter the thyroid gland. The iodide ions flow in the plasma, which is a large portion of the blood. This transporter uses the sodium electrochemical potential to drive the transport of one iodide ion and two sodium ions from the plasma into the follicular cells across the basolateral membrane. Now that the sodium has helped bring the iodide into the cell, its job is complete. Now, the sodium can leave the cell. This is done by the sodium-potassium ATPase. It transports 3 sodium ions out of the follicular cell back into the plasma and 2 potassium ions into the follicular cell. The next step is the movement of the iodide ion across the cell to the apical membrane, where it is transported to the colloid space via the Pendrin cotransporter. One iodide molecule is transported into the colloid space in exchange for 1 molecule of chloride into the follicular cell. Now our enzyme, thyroid peroxidase, comes into play. The exact 3D structure of thyroid peroxidase is not known as of yet. However, some laboratories have been working on solving this, and they have modeled possible structures of TPO. What is known about the structure of thyroid peroxidase is that it consists of 933 amino acids and that it has a large extracellular domain and a short transmembrane domain, which is inserted in the apical membrane of follicular cells. It is also known that TPO has 2 active sites, which function in both the oxidation of 2 iodide molecules to form iodine and the iodination of tyrosine residues. Therefore, while the structure of the active site is still unknown, we know that this enzyme has iodide as its substrate, and since iodide ions are negatively charged, we believe that there may be basic, or positively charged, amino acid residues within the active sites. TPO was synthesized in the ER of the follicular cell and secreted into the colloid space by exocytosis. Now that the iodide ions have entered the colloid space, in each of the 2 active sites of thyroid peroxidase, 1 ion of iodide enters. Thyroid peroxidase oxidizes the 2 iodide ions to form 1 iodine molecule. Now that we have the iodine molecule, a protein called thyroglobulin comes into action, and our enzyme is recycled. Thyroglobulin consists of many large repeating domains, containing many tyrosine residues in each of these domains. Thyroid peroxidase iodinates the tyrosine residues of the thyroglobulin protein. If the thyroid peroxidase only iodinates one tyrosine residue of a domain, this forms monoiodotyrosine. If two tyrosine residues of a domain are iodinated, this forms diiodotyrosine. Iodination of the entire thyroglobulin initiates the conjugation of neighboring residues by thyroid peroxidase. This conjugation results in three types of hormones, including T3 and T4. Via the endocytic receptor located in the apical membrane of follicular cells, the iodinated and conjugated thyroglobulin enters the cells via endocytosis. Lysosomes containing proteases fuse to the thyroglobulin containing vesicles. The proteases cleave the peptide bonds between the thyroid hormones and the thyroglobulin backbone. Now, the T3 and T4 molecules are free to be transported across the cell to the monocarboxylate transporter, or MCT. This transporter moves the hormones across the basolateral membrane to the plasma, where they can be transported for use throughout the body. Remember what we said earlier? Metabolism rate is adjusted when the amount of thyroid hormones being released is altered. Hyperthyroidism results when hormone levels are too high, while hypothyroidism results when hormone levels are too low. Both affect the body's metabolism and are often caused by autoimmune disorders. In individuals with an autoimmune disease, the body can produce proteins called TPO antibodies. Normally, antibodies protect against viruses and bacteria. However, in this case, these antibodies attack healthy tissues and organs, including the cells in the thyroid. As previously mentioned, autoimmune disorders are common causes of hyperthyroidism and hypothyroidism. Firstly, Hashimoto's disease, also referred to as thyroiditis, is a common autoimmune disease which causes hypothyroidism, despite being iodine sufficient. High concentrations of antibodies are detected in individuals with Hashimoto's disease. These antibodies, mainly TPO antibodies, attack the thyroid, causing inflammation and decreased production of the thyroid hormones. Furthermore, hypothyroidism can also be caused by iodine deficiencies. Another common autoimmune disease is Graves' disease, which causes hyperthyroidism. Graves' disease primarily affects the thyroid gland. It is caused by autoantibodies that bind to and stimulate the thyroid hormone receptor. These autoantibodies act as agonists and induce excessive thyroid hormone secretion. Hyperthyroidism can also be a result of excess iodine. An individual can be tested for an autoimmune disorder and other TPO-related diseases by taking a blood test called the TPO test. It detects any antibodies in the blood that attack TPO. A negative result means that there are no antibodies present in the blood, whereas a positive result means that there are antibodies present and indicates that an autoimmune condition is affecting the thyroid. Individuals with TPO antibodies have decreased thyroid hormone levels because the antibodies bind to and block TPO, the key enzyme involved in producing thyroid hormones. The TSH test measures the amount of TSH present in the blood. TSH stands for thyroid stimulating hormone. Too high or too low levels of TSH can indicate a thyroid problem. The thyroid is controlled by the pituitary gland, which produces TSH. TSH tells the thyroid how much thyroid hormone needs to be produced. The TSH test can be used to find out if an individual's thyroid is producing the correct amount of hormones, which indicates if a person has hyperthyroidism or hypothyroidism.